Hello friends, welcome to BMH Learning. In this video, we will discuss about Microbial Assay of Antibiotics by cup plate method. What is a microbial assay? A microbiological, or we can say microbial assay, is defined as qualitative or quantitative determination of chemical compound from a simple or even complex material with the use of microorganisms. Now, talking about the microbial assay of antibiotics. So this is basically used for demonstrating the therapeutic efficacy of antibiotics. That is, how much the given antibiotics are effective against specific bacteria in terms of preventing its growth. For microbial assay of antibiotics, two methods are generally employed. The cup plate, or also called cylinder plate method, and the turbidimetric or tube assay method. This tutorial will deal with cup plate method. So what happens in cup plate method? It depends upon diffusion of the antibiotic from a cylinder. Now, the cylinder is placed vertically in the solidified agar layer. The diffusion through the cylinder prevents the growth of the added microorganisms present around the cylinder, thus forming a zone of inhibition, the preparation of agar. So, according to the need of the experiment, required amount of agar solution is prepared. The agar solution is then poured into the petri dish. The important thing here is we inoculate microbial inoculum over the entire agar surface. So the agar solution has the bacteria against which we are testing the antibiotic. Next step is the perforation on media plate. So the media plate is perforated, or in simple words, we can say the wells are made with the help of a well puncher. You have to note one thing here, that the number of times we perforate the media plate or what number of wells we make actually depends on how many antibiotics we want to test. So, once the required number of wells are formed, the antibiotic solutions are added into their respective wells. The wells are then correctly marked. The plate is then kept for incubation at 37 degrees centigrade. Results and interpretation. The different antibiotics used are of varying efficacies. That means one antibiotic restricts the growth of bacteria more or less than the other antibiotic. Suppose antibiotics A and B form these kind of zones of inhibition. The diameter of the zone A is bigger than the diameter of zone B. This implies antibiotic A is more effective in preventing the growth of bacteria. Similarly, other antibiotics produce different zones of inhibition. Antibiotic D has very large diameter of zone ring, representing high efficacy antibiotic. And regarding antibiotic F, the diameter of inhibition zone is small, so this one is low efficacy antibiotic. This was all. Thanks for watching.